How do you know a new therapy actually works and is not just making people feel heard? How do you separate a real drug effect from a sugar pill? Experimental design is the answer, and it is harder than it looks. A hypothesis is a testable prediction about the relationship between variables. Good hypotheses are falsifiable, meaning evidence could potentially prove them wrong. A hypothesis that cannot be falsified is not scientific, no matter how true it might seem. The independent variable is the factor the researcher deliberately manipulates. The dependent variable is what the researcher measures as an outcome. A memory researcher might manipulate sleep hours as the independent variable and measure recall accuracy as the dependent variable. An operational definition specifies exactly how a variable is measured or manipulated. Hunger could be operationally defined as hours since last meal. Anxiety could be defined as score on a validated questionnaire. Without operational definitions, replication is impossible. Extraneous variables are factors other than the independent variable that could affect the dependent variable. When they differ systematically between groups, they become confounding variables, making it impossible to determine whether the independent variable actually caused the result. A theory is a well-substantiated explanation supported by repeated testing. In science, a theory is the strongest form of explanation, more powerful than a hypothesis, built from years of converging evidence. The word theory in science does not mean guess. A control group receives no treatment or the standard treatment, providing a baseline for comparison. Without a control group, a researcher cannot determine whether the treatment caused changes or whether participants simply improved over time on their own. Random assignment means each participant has an equal chance of being placed in any condition. It distributes pre-existing differences between groups by chance, equating groups on all variables except the independent variable before the study begins. Random selection means choosing participants randomly from a population, which improves how well results generalize. Random assignment means randomly placing participants into conditions, which improves how confidently we conclude causation. These are two different procedures with different goals. The placebo effect occurs when participants improve simply because they believe they are receiving treatment. It accounts for 30 to 50% of improvement in many drug trials. Placebos trigger real biological responses, including endorphin release and measurable changes in brain activity. In a single-blind study, participants do not know whether they receive the real treatment or a placebo. This prevents participant expectations from biasing reported outcomes. However, researchers who know group assignments may still unconsciously behave differently toward each group. In a double-blind study, neither the participants nor the researchers interacting with them know who received which condition. This prevents both participant expectation and experimenter bias from influencing results. Double-blind trials are the gold standard in drug research. Demand characteristics occur when participants figure out the purpose of a study and alter their behavior accordingly. They may try to confirm the hypothesis or appear favorable to the researcher. This contamination makes the study measure participants guessing, not the actual variable. Social desirability bias occurs when participants answer in ways that seem socially acceptable rather than honestly. It is especially problematic in surveys about drug use, prejudice, or illegal activity. Anonymity reduces this bias, but cannot eliminate it entirely. Selection bias occurs when the sample does not represent the population. The weird problem means most psychology research uses Western, educated, industrialized, rich, democratic participants, limiting how well findings generalize to the rest of the world's population. Replication means repeating a study to see whether the same results emerge. It is the cornerstone of science. A single study can be wrong by chance, by fraud, or by error. Replicated results across multiple labs and samples are far more trustworthy. The replication crisis emerged when researchers attempted to replicate 100 published psychology studies in 2011. Only about 36% replicated successfully. This finding revealed serious problems with how psychology research was conducted and reported, prompting major reforms. Publication bias means journals prefer positive results over null results. This creates the file drawer problem. Studies that found no effect stay unpublished in researchers' drawers, while only positive findings get published, making treatments appear more effective than they really are. Meta-analysis combines data from many studies on the same topic to compute an overall effect size. It is more powerful than any single study because it encompasses thousands of participants across many settings. Meta-analysis is now considered the strongest form of evidence in psychology. Peer review is the process by which scientists evaluate each other's work before publication. Reviewers examine methodology, statistics, and conclusions. While imperfect, peer review filters out major errors and is considered the minimum quality standard for published psychological research.